Traveler's diarrhea is a significant health burden to children under 5 years of age, travelers, and deployed service personnel in areas where ETEC bacteria that cause the disease are endemic. NIGMS-funded basic biomedical research has led to vaccine development against the bacteria that cause this diarrhea. My lab studies long, thin fibers on the bacteria's surface that you see here on the left that are essential for binding of bacteria to the small intestines, where they then initiate disease. In collaboration with Stephen Savarino's lab and Dee Shah's lab, we have determined the structure of these pili or fimbriae, shown on the right. Why is this important? Bacteria bind to the epithelial cells of the small intestines using pili, or these long filaments, and unless they're washed away, they can stay bound and initiate disease. We found that the long fibers are helical, and as you can see, a small portion of the filament from the blue box, the structure is shown on the right. The pink protein is a tip protein that binds particularly to the target epithelial cell, and then there are about 1,000 copies of the major subunit, which is shown in yellow and blue. We showed that the helical filament at the black arrow can also unwind, shown by the blue arrow, under fluid flow, so that allows the bacteria to distribute the force more evenly and not get washed away. So it's a good strategy that the bacterium uses. Wild-type bacteria have both helical and unwound pili, but what happens when there's a mutation and you can only have unwound pili? In the top row, wild-type bacteria can clump red blood cells, and the bottom row, they do not clump when there's no tip adhesion to bind. In the middle row, unwound pili appear to begin clumping the red blood cells, but then over time they go back to being unclumped, showing that the pili are not functional. We made a model of the unwound pili. Here's what that looks like, each subunit in a different color, like links on a chain. Dr. Shah's lab did an X-ray crystallography structure to show this in more detail, and what you can see is a tongue and groove arrangement, which makes the filament very strong. Pillin-based vaccines had not worked in the past. We decided to go back to our structure to try and understand why. What we found was that closely related strains of the bacteria had pili that were extremely different on the surface. So the red shows what is different, and the white is some change, and the blue is very conserved. And what we saw was that it was not going to be possible to make a pillin-based vaccine that had broad coverage. Based on the results of our research, Captain Savarino is now using the tip adhesion, shown in pink here, as the basis for his vaccine that is currently in phase 2B trials. Thus, fundamental biomedical research has provided the basis for development of a vaccine against traveler's diarrhea, which can improve health worldwide.